Okay, there we are. I'm recording. Let me share screen. Okay, so here's the lab paper. Hope everybody has a copy of this. And this is what you'd be turning in. You don't have to turn the whole thing in, just the last pages where you need to answer questions, and then turn it in to me. And then I grade it and give you a grade for it. All right? Okay, and interrupt me at any time. It doesn't matter. If you got a question, interrupt me. And I'll ask you some questions and expect someone to answer. Okay, so getting ready. And usually these getting ready things in these, I don't usually have you fill those out or, at the beginning of each class, I tend to go over it myself as part of the introduction to the lab. And part of this will be stuff on the next exam, because the next exam is going to be dealing with cells and working cell, which is partially cell transport, which
it's
is
in my pocket upstairs.
water
I
in
didn't
solutions.
even know it was gone until yesterday, but I thought I'd lost it.
Skylar, could you mute if you're going to be talking to somebody? I assume it's Skylar because your microphone's on. I can hear you.
Oh.
Okay, thanks. Okay, so unmute if you want to ask a question, obviously. Okay, but it's tough for me to talk when there's other noise in the background. Okay, I assume you could do this, write a chemical formula of a water molecule. We did that in the molecular models lab, and draw a stick figure of a water molecule showing the position of each atom, right? And then label the type of bond between the atoms, and that's going to be a polar covalent bond. Right? Between the oxygen and the hydrogen is polar covalent. 2, draw two water molecules as they would be arranged together. Label the type of bond between these two water molecules. Oh, somebody's in the waiting room. Hang on. Now that's a little different. This is a different type of bond. Now this is a bond between molecules rather inside a molecule. Now later on, it's called a hydrogen bond. And later on, we'll see, when we talk about the larger macromolecules like nucleic acids and proteins, they are held together by hydrogen bonds, internal hydrogen bonds. Right? So the larger molecules are internal hydrogen bonds, but these hydrogen bonds are between the molecules, not within the molecule. Okay, and then the way they would be arranged would be, remember, water has an oxygen side and a hydrogen side, right? The oxygen bound to the two hydrogens. Well, the oxygen side has what's called a partial negative charge, because the oxygen is pulling the electrons, remember, closer to it and farther away from the hydrogen. That's the nature of a polar covalent bond, remember, unequal sharing. And electrons are negative, so the oxygen side of the molecule has a partial negative. The hydrogen side of the molecule has a partial positive, because it's a sort of like a lack of electrons. And what is that hydrogen out there is a proton, right? That's what a hydrogen atom is, a proton with one electron. Well, electrons being tried to take it away, so now there's a proton left over and it's positive. So the positive part of one molecule will be attracted to the negative part of another water molecule. As a matter of fact, each water molecule will be attracted to several other water molecules with hydrogen bonds. So every water molecule will be bound to several other water molecules by hydrogen bonds. And they're forming and breaking all the time, depending upon temperature, actually, which we'll get to. So these bonds, hydrogen bonds, remember, are the weakest bonds going, the weakest ones. But there's just so many of them in even the smallest drop of water that it really gives water this cohesiveness, which means that cohesive means the molecules are attracted to each other. So it's very cohesive. And it's also adhesive. It says that it will be attracted to anything else with a positive or negative charge.
Pay attention to that. He uploads documents, does he not? Would you calm down for 2 seconds? Take a couple of deep breaths and calm down. It's not how you deal with things.
Who's talking? Emily? Okay. Okay, so then we're going to look at how these what these hydrogen bonds look like, but then define the following terms. And we've already defined a lot of these, but they're still important to understand. And some of them are going to be a little newer. Just to define two of them. We know what well, let me point to it. I keep on pointing my hand. You can't see my hand. I got to use the cursor. Covalent bond. We know what a covalent bond. Polar molecule. We know what a polar molecule is. It's got oxygen in it or nitrogen, and it's got polar covalent bonds. Ion. You know what an ion is? An atom that has given up or taken on an electron. You know what an ionic bond is? One atom gives up an electron, and it makes a deal with another atom that gains an electron. Right. Aqueous solution. This is a water-based solution. A solution is a mixture of at least two molecules, where one is dissolved in the other. Right. And then these next two, so you got to know these next two terms, solvent and solute.
Excuse me.
The solute is the molecule that's dissolved. The solvent is a molecule doing the dissolving. See? So the biological solvent is water. And any solution where water is the solvent is called an aqueous solution.
Mr. Murphy,
Yes.
Adrienne says that she's in the waiting room.
Oh, good, thanks. I didn't see, usually people, there's a little blurb comes up when people are in the waiting room. I don't see it. So let me go, I think I got to stop screen sharing here. Let me grab her. Stop screen sharing. See, I don't see him in a waiting room. So I'm gonna have to go to send her an invite. Adrienne Grove, right? Okay, I'm sending her an invite.
Okay, thank you.
Thanks for letting me know. I don't know why I can't see her in the waiting room, but I assume she'll get in here in a minute or so. Okay, let's go back. Screen share. Okay, and all of your precious body fluids, they're almost all aqueous. So it's about blood or interstitial fluid or lymph, it's all aqueous. There's only a couple that are nonpolar, like sebaceous glands in your skin, put out this oily solution. Cereminous glands in your ear, they make earwax. That's a nonpolar. But all the rest of your body fluids are aqueous. Okay, and so your blood, for example, is blood plasma is a complex solution of water as a solvent and lots of solutes, ions, nutrients, waste products, signaling molecules, plasma proteins. They're all dissolved. Okay. So something that's capable of being dissolved is called soluble. So is sugar soluble in water? Yeah. Right. Insoluble is something that's not soluble in that particular solvent. Like, oh, okay, there's Adrienne right there. Like oil in water. Oil is not soluble, it's insoluble in an aqueous solution. And these next terms, J and K, remember, they're the same as polar and nonpolar. Hydrophilic, water-loving, polar; hydrophobic, water-fearing, nonpolar. And we just mentioned what a hydrogen bond was, and I defined cohesion and adhesion. And now we're going to look at one property property of any fluid is surface tension. That is the force you need to push on to put something into water. Like you have a bowl full of water, you want to put your finger in it, you got to break the surface tension to do that. Not much. And it seems like, it's not much, but actually there are so many hydrogen bonds in the surface of water that if you spread out the weight enough, you can float incredibly heavy things, right? Like an aircraft carrier. What's holding up an aircraft carrier? Hydrogen bonds. That's what's holding it up. You spread it out enough, you can float an aircraft carrier. So it's really significant, right? And density is how close molecules are together in a substance. And usually, the colder something is, the more dense it is. The warmer something is, the less dense it is, because of molecular movement. Remember, molecules are always moving at any temperature except absolute zero, which doesn't exist. Molecules move, and the hotter they are, the more violently they move, and the farther apart they get. The colder they are, the less they move, and the closer together they get. Except for water. Water has this bizarre property we're going to talk about when we're talking about making ice. It's really bizarre. We're going to talk about it. Okay. So then we get to general overview. So water is essential for life. It's the biological solvent. Most abundant inorganic molecule in most cells. And most molecules make up the cell are dissolved in this water. And all the biochemical reactions necessary to sustain life occur in a solution of water. And life probably began in water. And so when NASA sends out rockets to the different planets looking for life, send out probes looking for life, what are they looking for? They're looking for water. Not only water, but water in a liquid form. So they look for, so-called habitable planet will be a planet that's in what's called the Goldilocks zone. That is, it's not too cold, so if any water is present, it's ice. It's not too hot, so any water that's present is steam. It's just at the right temperature. Porridge is not too cold, not too hot. We have liquid water. That's called the habitable zone or the Goldilocks zone. Okay. So to understand cells, organisms, life process, it's necessary to first understand the physical properties of water and how important these physical properties of water to life on Earth. Right? Chemical formula of water, familiar to all of us? Okay. It talks about, again, the polar covalent bonds, the partial negative charge, partial positive charge. There's the partial charges there, partial negative on the oxygen side, partial positive on the hydrogen side. All right. So they're attracted to each other by hydrogen bonds. Here are the hydrogen bonds. These little dotted red lines are the hydrogen bonds. So the oxygen side of one hydrogen water molecule is connected to the hydrogen side of another water molecule. It might affect several others. See how each water molecule makes several other hydrogen bonds with other water molecules. That makes it really cohesive. Okay. Okay, so like I said, even though individual hydrogen bonds are really weak, they account for most of water's most amazing properties. Okay, first of all, what we do is a really simple procedure that you just do and you notice what happened is the polar nature of water. How can you show that water is a polar molecule? Well, fill a burette with water. A burette is a long glass tube with a valve at one end. It's usually graded, so you know how much is in there. You fill that up with water and you have the valve closed. You put it over a beaker. You take a glass rod or better even a plastic rod, and we give you a piece of felt, and you'd rub the rod with the felt. And in doing so, you would impart a negative charge to it. So then you open up the valve on a burette and you let the water flow into the beaker. And you bring the rod close to where the water is leaving the burette. And what happens is you'll see the water will move toward the rod. It'll be attracted to the rod. So this is evidence that water is a polar molecule because you made a polar rod and the water is attracted to it. So it must be the hydrogen parts of the water molecules that are being attracted to the rod because the rod has become negative by rubbing it. If it was repelled by the rod, you'd also say water was polar, but it's never repelled, it's always attracted. Okay. So cohesive cohesion and surface tension. Okay, there's the ability of any substance to stick to itself or stick together, like Velcro. Okay, a drop of water will appear to have a skin which resists penetration, and also water beads up. It has nice round and it beads up because of these hydrogen bonds. And detergents, soaps, are special molecules that have both polar and non-polar characteristics. That's what all soaps are. They were called amphipathic molecules. They have they're polar on one side and non-polar on the other side, like a fatty acid. Polar on one side, non-polar on the other side. So they can mix with both. And that's how soaps work. Because dirt on your skin or dirt on clothes gets mixed with this oily sebaceous secretion on your skin. So in order to get to the dirt, you have to have something that's soluble in the oil, and a soap is. Soaps are made by taking usually animal fat and take it to a rendering plant, it's called. You don't want to live close by to one because they really stink. And they add these big polar groups to these fat molecules, and that makes a soap. So most soap is, animal-based. So a lot of people who don't want to use animals, you can get plant-based soap that's made from plant fat, but most soaps are made from animal fat. And they break hydrogen bonds. And so you want to solubilize the dirt that's in the oil, but then you want to wash it away with water. So you gotta have a molecule that both is soluble in oil and can be washed away with water. And soaps fit the bill. That's why soap dries out your skin, because it takes away the oil that makes your skin supple. Okay. Obtain a clean microscope slide, put a drop of water on one end, drop of alcohol on the other. Observe the two drops and record your observations. And we're going to be talking about that later. And you got to remember the structure of alcohol. Alcohol is cohesive also, but not nearly as much. So when we get the analysis results, we'll talk about what you see when you do that. Then you float a pin. You take a dish of water, you put a piece of tissue paper down, put the pin on the tissue paper, take two toothpicks, and you push the tissue away, and you'll float that pin. And you'll see that the water is like bent around this pin that's floating. Then you'll add a drop of detergent, and you'll see what happens. Pin drops like a stone right to the bottom, because the detergent, it's hydrogen bonds that are holding up that pin. So you break those hydrogen bonds, the surface tension of water decreases, and there you go, the pin falls. Okay, water as a solvent. So like I said, in aqueous solutions, water is the solvent. The material dissolved is the solute. And okay, so a molecule which forms a solution in water is soluble or hydrophilic. And all salts are soluble because ionic bonds, they break in water. They always break in water. Right? And so you'll see here spheres of hydration form around salt ions. So you have the sodium chloride, and the water gets in between those, and they surround the oxygen part of water molecules will surround the sodium ion because it's positively charged. And then the hydrogen part of water molecules will surround the chloride ion because it's negatively charged. And this is how water dissolves salt. Okay. But also other polar molecules that have polar covalent bonds will also be soluble in water. So in the ionic compounds and compounds that have polar covalent bonds are soluble in water. Insoluble molecules are nonpolar, not form a solution. They can't form hydrogen bonds with water. They can't even find any kind of electrical interaction with water. So they're insoluble in water, hydrophobic or water-fearing. Okay. And so what you'll do is, what you would have done in lab is, you're given these 50 milliliter test tubes. You fill one with water. Oh, you have water, and then you take these substances, water with rich food coloring, so you can see it. You add it to the water, and see if it dissolves. Obviously, it's going to dissolve. Then you add salt and see if it dissolves. Sucrose, ethanol, and mineral oil. And you shake each one and see whether it dissolves. This is a way you can actually determine whether the substance that you have is polar or not. You may not know its chemical formula. Now, if you know its chemical formula, how do you tell whether it's polar? Well, look for oxygen and nitrogen. If it's got lots of oxygen and nitrogen in it, it's going to be polar. But what if you have like this powder in lab and you don't know whether it's polar or not? Put it in water. Does it dissolve in water? It's polar. If it doesn't, it's non-polar. Simple as that. So we're going to find out which substances here are polar, which ones are not. Okay. Acids, bases, buffers, and pH. Okay. Some materials, when dissolved in water, have the ability to change the pH of a solution. Okay. Remember, when I want to change the pH of the solution, the ionic compounds must contain either protons, hydrogen ions, or hydroxide, OH ions, because that's what pH measures. pH measures the amount of hydrogen ion in a solution versus the amount of hydroxide ion in a solution. It's a logarithmic scale from 0 to 14. pH of pure water is 7. Solutions with pH between 0 and 7 are acidic. Between 7 and 14 are basic. So what pH 7 means is that then the concentration of hydrogen ions is exactly the same as the concentration of hydroxide. That's pH 7, that's neutral. And that's what the pH of distilled water is. Because every aqueous solution has got a pH. Every aqueous solution is going to have hydrogen ions and hydroxide ions. Why? Because of the ionization of water. Water breaks apart. Let me go and show you that. Stop sharing here. And let's go on the web. Wait a minute. Let's go on the web. Let's go to Google. Okay, the ionization of water. Um, okay, where is it? Okay, you get, well, don't worry about all these equations done, but just look at this thing. H2O breaks up the hydrogen ion and hydroxide ion. So as I mentioned before, oxygen loves electrons and tries to steal electrons away from other atoms. And sometimes it succeeds and the molecule breaks apart. And that's what happens to water. That, wait a minute, maybe this is better. Yeah, right there. Don't worry about this KW, I don't know what that is. But the water breaks up into those two ions. And remember, that's a reversible reaction. So it goes back and forth. And so in even the smallest drop of water, you've got lots of water molecules breaking apart, reforming, breaking apart, reforming. So there's if it's an aqueous solution, it's got a pH, all right? Okay, there's the pH scale. So it's 0 to 14, 1 to 14. And here's neutral pH, and that's pure water. And down here is acid. That means the hydrogen ion concentration is higher than the hydroxide concentration. And it's a logarithmic scale. Now, don't worry, I'm never going to give you logs or anything. But so that at pH 6, there are 10 times the hydrogen ions as in pH 7. pH 5 has 100 times the higher hydrogen ions. pH 4 has 1,000 times, and so on and so forth. So even small changes in pH mean a big change in the concentrations of these ions, okay? Now up here is basic or alkaline, and the concentration of hydroxides is much higher than hydrogen ions. And again, it's logarithmic. Every pH unit means is 10 times more hydroxide ions than hydrogen ions. So why is this important to living things? Well, hydrogen ions and hydroxide ions are very reactive things because they have full charges. So they're really reactive, and they can really affect the shape of molecules, the chemical reactions going on. It can really disrupt things. So organisms have evolved to have a pH somewhere near neutrality, and they have to maintain it. It's a homeostatic variable. You have to maintain like your blood pH. If it gets off even a little bit, you are in serious trouble, right? And so our doesn't really have the blood. Here it is. So blood is slightly on the alkaline side. It's 7.4. Your blood is 7.4. And that's true for pretty much every animal, as their body fluids are 7.4. And they really got to stay there. Now, and you can see all these things down here are even lemon juice is down to 2. But when you get down to stomach acid and HCl, wow, they become really dangerous. If you get really, really acidic things, they can become actually really dangerous. Because HCl is what's called a strong acid. Why is it strong? Well, what's the bond between hydrogen and chloride? HCl, hydrochloric acid. What is the nature of the bond between the hydrogen and the chloride and the chlorine? Anybody? What kind of bond is it? To give you a hint, what column is hydrogen in? First column, right? in the periodic table. What column is chlorine in? 7th. What do atoms in the first column make with atoms in the 7th column? What type of bond? You got to know for this first exam. You got to know what kind of bond do they make. How many valence electrons does hydrogen have? It's in the first column. Anybody want to venture?
Is it covalent?
No, it's not covalent. Well, let's look at it this way. Hydrogen, how many valence electrons does hydrogen have?
Would it be 1?
It's 1. It's in column 1. That 1. All right. So it really wants to what? Get rid of that 1. How many valence electrons does chlorine have? It's in column 7.
Would that mean it has 7?
7. It wants 1 more to fill up its valence shell. So it will accept an electron from hydrogen to form hydrochloric acid. So what kind of bond is that? Well, it's not covalent, not hydrogen. What kind of bond is it?
Ionic?
It's ionic bond. And what do you know about ionic bonds when you put them into water? They break apart 100%. So an acid is something that adds hydrogen ions to a solution. You put HCl into water, what are you going to get? Lots of hydrogen ions because it's going to break away from the chloride ion. So that makes it a strong acid. As opposed to a weak acid, which would be like organic acids, like acetic acid. Let me pull that up. Okay, this is an acid. Well, we know that the carboxyl group is an acidic group, right? Why? Well, this bond here, well, they don't put it in, but you know, there's a bond between that oxygen and that hydrogen. That breaks sometimes. And the hydrogen ion takes off, and you get and that has a positive charge, and then the rest of the molecule becomes this big negative ion. And does it show breaking off? No. So that means this is an acid, but it's a weak acid because it doesn't break 100%. So, a certain percentage of the molecules break and produce hydrogen ions, and other ones get back together, and it's not 100%. So that's the difference between a weak acid and a strong acid. Okay. Let me go back to pH scale. And then you go up here to baking soda, milk of magnesia, ammonia, soap, bleach, drain cleaner. This is why you gotta be careful with drain cleaner. So maybe you want to wear gloves or something when you're cleaning your drains because it's really, really basic. And it's just as dangerous as being very, very acidic. And what we use in class for a base is sodium hydroxide. NaOH. Now, here's the thing with NaOH, this is more like it. The bond between the oxygen and hydrogen is polar covalent, but the bond between sodium and the oxygen is actually ionic. So here's an exception to the rule. Like I said, the guys in column 1 and column 2 always make ionic bonds with the guys in column 7. But occasionally usually the atoms in columns 3 through 6 make covalent bonds. But they occasionally make ionic too. Oxygen sometimes make an ionic bond. So this is an ionic bond. That's why it has this dotted line here rather than a straight line. And so, and what do you know about ionic bonds? You put it in water, they break apart. And this thing goes one way, this thing goes the other way, hydroxide ion, which is basic and will raise the pH. Right? So is that clear, everybody? So we go back to here. Okay. Oh, and the last thing, pH is so important to you that that is what determines how often you're breathing right now. You're breathing about 12 times per minute. Why? Why 12 times? Well, it's to maintain your blood pH. Most people think it's to get oxygen. Well, you get oxygen that way, but oxygen has almost nothing to do with how often you breathe. It's pH. It's much more important to regulate how much CO2 you're getting rid of rather than how much oxygen you're getting in. It seems contradictory because CO2 is a waste product, but it's really closely tied to pH. There's an equation that shows that, but it's suffice it to say it's really closely tied to pH. The more CO2 in your blood, the more alkaline your blood will be. No, the more acidic your blood will be. The less CO2 in your blood, the more alkaline your blood will be. So you are breathing at the right rate to get rid of just the right amount of CO2 to keep your blood pH at 7.4. You can voluntarily screw that up if you want to by hyperventilating. Why do I feel dizzy now? Well, I just screwed up my blood pH voluntarily. I get rid of CO2 too rapidly, which means that my blood pH went up. It's called respiratory alkalosis. And now there's a pH regulator in your brain that will tell your diaphragm, stop, slow down the breathing. You're getting rid of too much CO2. Or if you hold your breath, your blood will eventually become more and more acidic. And that's why you'll have to breathe more. Once you stop holding your breath, you have to hyperventilate a little bit. Okay, now, buffers, right down here. They're really important because there are compounds coming into your blood and leaving your blood that are acids and bases. And you can't let your blood pH change by very much. So so a buffer is a compound that minimizes pH changes. And the buffer can be in water, and the water can still be 7.0. And so what they are compounds that can donate hydrogen ions to a solution, or soak up hydrogen ions, depending upon how many hydrogen ions there are. I'll give you an example. You have a beaker full of distilled water, you add a drop of hydrochloric acid. Boom, you go down to pH 3 or 4. What if you have a water with a buffer in it, and you're at pH 7, you add a drop of hydrochloric acid, you drop to 6.8 or 6.7 or something. You drop a fraction of a pH rather than several pH units. So it minimizes the change. And same thing with sodium hydroxide. You have beaker full of distilled water, you put a drop of sodium hydroxide, boom, up to pH 10 or something. If the buffer is there, the pH goes up to 7.2, 7.4, 7.6 or something, or 7.7 rather than 10. And so it'll be a non-lethal change. So you need that because you have you're constantly making metabolic acids. Most people do. And you have to get rid of that acid. And the way you do it is peeing. You pee. If you test the pH of urine, it's almost always acidic because most people have an acid load. Some people who are vegans actually have a basic load, but most of us eat meat and eat regular foods, and so we make acid, and we can't help it. It's put into the blood, it's carried to the kidneys get rid of it. Now, and it's carried in the blood, and it's buffered. So your blood is loaded with buffers to make sure that when that acid leaves cells and is brought to the kidneys, doesn't change the pH of your blood very much. Okay, so buffers are required for living things. Okay. Okay. And salts are also highly soluble, but if the salt doesn't have hydrogen ions or hydroxide ions in it, it doesn't affect the pH of a solution. Okay. So here's the procedure. You have water with buffer in it. You have HCl, NaOH, and NaCl. So we're going to fill out a table later on about what happens to pH when you add HCl, NaOH, or NaCl into water without buffer and water with buffer. Okay. Water temperature and density. We mentioned density, a measure of the mass or number of molecules in a unit of volume. Inverse relationship between density and temperature for most substances. That is, the higher the temperature, the lower the density. Okay. Okay. So as water cools, more and more hydrogen bonds will form between the molecules. So the closer the water molecules get to each other, the more hydrogen bonds they form. The farther apart they get, the fewer hydrogen bonds they make. Right? So that's a direct relationship between temperature of water and how many hydrogen bonds it makes. The hotter it is, the fewer hydrogen bonds. Okay. Okay, so, and we're going to look at the change of the density of water as a function of temperature. How does temperature affect the density of water? And it affects it in a really bizarre way. It turns out that water is most dense at 4 degrees C, not 0. When water cools down to from like 22 degrees room temperature to 4, it gets denser and denser and denser and denser. But if it cools down below 4, it gets less dense and less dense and less dense until it finally freezes. And in ice, the water molecules are farther apart than they are in water at 22 degrees. And so that's why, as we all know, ice floats. Even though it's colder, and even though it's a solid, it floats in water because it's less dense. It's bizarre. It's a bizarre property of water. Okay. So how we show this is you fill a beaker with 800 milliliter warm water. And then we take this water from the refrigerator that has red dye in it, and we put it in a pipette, and we put the pipette just below the surface of water, and we slowly release the cold red water into the warm water, and it drops right to the bottom because it's denser. And then as it warms up from the bottom, it starts to rise up, and then it makes the whole beaker red. Then we get some blue ice cubes that has a blue color in it, and we drop them in the warm water, and of course the ice floats because it's less dense. And as it melts, you can see the blue dropping down as it melts and warms up from 0 to 1 degree, 2 degree. It drops right down to the bottom. Because when it gets to 4 degrees, it's most dense. And then it will warm up more and start becoming less dense, and it'll turn the whole beaker blue. Okay. Come on, get rid of that. Okay, cell size and diffusion. Oh my God, okay. Organisms vary greatly in size from microscopic plankton to blue whales. So when we look at the individual cells that make up these organisms, how do you compare the size of a cell in the plankton versus the size of a cell in the blue whale? Are the blue whale cells bigger than the plankton cells? Or are they the same size? Or are the plankton cells bigger than the blue whale cells? Okay, it's in the next sentence. We find that the cells of plankton and cells of blue, they're all microscopic. They're all really tiny. They're the same size. It's just the blue whale has a lot more of them, right? So cells have to be small, microscopic. And that is because they have to make sure that the rate of diffusion is fast. So you have to think about things that affect the rate of diffusion. And things like temperature affects the rate of diffusion. Faster, the warmer things are, the faster things diffuse. The distance that things are diffusing, that affects it. The farther apart they are, the slower diffusion is. And what's called the surface area to volume ratio affects diffusion. Let me explain that. So what you'd be doing in lab would be you'd be getting this gelatin that has a cloudy color because it's been infused with this phenolphthalein. It's a pH indicator. When it's a pH 7, it's milky color. When it pH rises into the basic range, it turns pink. Okay, so you cut out like a 3 centimeter cube, a cube of gelatin that's 3 centimeters on the side, and you cut out a cube that's a 0.5 centimeter on the side. You both put them in. You put them into this this bowl, and you pour sodium hydroxide over the top. You cover them with sodium hydroxide for a few minutes. What happens? The outside becomes pink. You leave them in there for a few minutes. You take them out. You take a scalpel and you slice them in half. You flay them open and look to see inside. Well, what you find out is that the pink color is all the way through the small cube. But in the big cube, it's still milky on the inside. It's just a thin pink layer on the outside. So that demonstrates that the diffusion was happening slower in the big cube than in the smaller cube. And why is that? That is because of what's called the surface area to volume ratio. Let's look at that. So surface area is the amount of area around the cube. Right? Volume is the amount of stuff inside the cube. Right? So how do you measure surface area? Well, if you have a side of a cube, so S is the length or width of a cube. Remember, a cube has got the same width, length, and depth. That's a cube, and that will be S. So the surface area of one side of the cube is the length times the height. S times S, S squared. Okay, that's one side of a cube. How many sides does a cube have? 6. So the entire surface area is 6s cubed. Squared, 6s2, superscript 2, 6s squared. But how do you measure volume? Well, volume is the height, the length, and the depth. So S times S times S. So S cubed. All right. So here's the thing. As you go from a small cube to a big cube, what happens to the surface area? Stay the same? Does it get larger? Does it get smaller? As you go from a small cube to a big cube, the surface area go up? Well, yes, S is larger. So the surface area goes up. How about the volume? Does it go up? Well, yeah, S is larger. So there's more volume in a big cube than there is in a small cube. So they both go up. That's important to remember. But what about the relationship between the two? Between the surface area and the volume? That is the surface area to volume ratio, surface area divided by volume. What happens to that number as you go from a small cube to a big cube? That number goes down. Why? Remember, 6s squared is in the numerator, right? And as you go from small to large cube, that gets to be a bigger number. The s cubed, the volume is in the denominator. So as you go from small to bigger cube, that gets larger. Also, but it gets larger faster than the surface area because it's s cubed for the volume and s squared for the surface area. So as you get from smaller, and so you know, and with a fraction, the bigger the denominator, the smaller the number. The bigger the numerator, the bigger the number. The bigger the denominator, the smaller the number. So the volume increases faster than the surface area. So as you go from smaller to bigger and bigger cubes, the SA over V, surface area to volume ratio, gets smaller and smaller and smaller. And that means that the diffusion becomes slower and slower and slower. So if you had a cell, a really big cell, diffusion would be so slow, you couldn't get oxygen in fast enough or nutrients in fast enough or get waste products away fast enough to keep that cell alive. So cells have to be small. And that's why they're small. And you can show that with this experiment with the gelatin. Any questions about that? Okay, before we start the analysis of results, there's one other really important property of water that's not, that we used to do an experiment with, but I guess we don't do it anymore, and that's what's called the specific heat of water. And this is an incredibly important property of water that keeps us alive on Earth, and probably if it didn't have this, there wouldn't be any life on Earth. Water has a really incredible ability to store heat. It takes a lot to heat water up, but it takes a lot and then water releases the heat when it cools down. As a matter of fact, that's also true for how we cool ourselves. We cool ourselves with the evaporation of water. When water evaporates, it takes away lots of heat. That's why we have a great thermoregulatory system. When we get too hot, we sweat, and that evaporative cooling takes heat away. But also, water, like I said, it takes a lot to heat it up, but it really holds on to that heat. eventually gives it up, but it hangs onto it and then releases it slowly. This allows life on Earth because what's the difference between temperature between night and day on Earth? It's like 30 degrees out here. Maybe get down to 25 tonight. It's like 5-6 degrees difference between day and night, right? When the sun is gone. Why is that? It's because we call this planet Earth, but it's mostly water. 70% of the planet is covered by water, and that's a huge heat sink. I mean, that holds tremendous amount of heat. So during the day, it's warmed up by sunlight, and at night, when the sun is gone, there's somebody in the waiting. When the sun is gone, the heat comes out of the oceans and big bodies of water and spreads over the land and minimizes the temperature differences between night and day. If you want a planet that didn't have that huge amounts of water, the difference between night and day would be hundreds of degrees. But it's not, because we're a water planet. So, and that's why actually it's bigger difference between night and day, and differences in temperature in the seasons in the middle of large continents where they're really far away from, the ocean. There's much bigger changes in the temperature between night and day, and between summer and winter. You go out to North Dakota, you're roasting in the summer and you're freezing in the winter. So, and actually, the temperatures in the U.S. are mitigated by the Gulf of Mexico coming up there, and so lots of the country are really close to water, and so we have smaller temperature changes. Okay. So let's answer these questions now. All right, the analysis of results. Where are we here? Hey, how come I'm not moving? What's going on? Something's wrong with my, hang on a second. What's the problem here? Let's try it again. Okay, so let's analyze the results, answer these questions. Now, one thing, some of these questions ask you to draw stuff. I don't care if you draw, we don't really do them, so you don't have to draw. So it's first one, you don't have to draw the burette. There's a couple other ones that would ask you to draw. I'm gonna say, forget that, except there's a couple of thumbnails drawings for graphs later on that you should do. But this first one, don't worry about it. Which part of the water molecule is drawn toward a negatively charged rod? The hydrogen sign, because that's the positive sign. Which is repelled by the rod. If it was repelled, it'd be the oxygen side, because that's negative. But it's never repelled. It's always attracted. All right. Cohesion and surface tension. So relative diameter of one drop. Well, it's smaller in water, larger in alcohol. If you put a drop of water on a glass slide, it beads up. It's nice and round and a nice bead. You put alcohol on it, it slides all over the place and forms this very flat, irregular splash. So it's larger. And this is because, remember the structure of alcohol, it has a hydroxyl group. So it has polar covalent bond, but only on one side of the molecule, not on the other side. So it really only forms like one hydrogen bond. Whereas water is as polar on both sides and forms several hydrogen bonds. So it's much more cohesive. So the elevation of height of one drop, tall for water, short for alcohol. Appearance of margin edges, regular for water, nice and round, irregular for alcohol. And then you wait to see which one evaporates first. You don't have to wait till both of them evaporate, but which one evaporates first? Alcohol. Because all it has to do is break one hydrogen bond and it takes off. Where water has to break several, each water molecule has to break several hydrogen bonds before it can take off and evaporate. And remember, evaporation takes away heat. So that's why you put alcohol on your skin, you let it evaporate, you feel coolness, because it takes away heat when it evaporates also. Okay. So this is so relative rate of evaporation, slow for water, fast for alcohol. Then, comparison of cohesive properties of water and alcohol. Demonstrate you understand cohesion. Explain why the drops of alcohol and water behave differently, because the water makes many, many more hydrogen bonds than the alcohol does. And that's why it has those characteristics. Okay, another drawing, question 2 here. Draw a picture of the floating pin. No, don't worry about it. But if you do float a pin, you can see that the water is bent around the pin. You can actually see it dipping in and bending. Explain what a pin floats. In other words, is the force of surface tension greater than or less than the force of gravity at that point? This is question 3. Which one's greater when the pin is floating? Force of surface tension or gravity?
Surface tension.
Yeah, because it's floating. That's correct. So it's being pulled down by gravity, but it's being held up by surface tension. So surface tension must be stronger. Why the pin sinks when the detergent is added culture? Because it breaks hydrogen bonds. It disrupts hydrogen bonds. And that's what's holding the pin up. So we add detergent to our laundry and water used to wash dishes because there's oily stuff on the dishes. And we need something that will solubilize the oily stuff. And then we want to wash away with water. So we're going to have something that mixes with oil and with water. An amphipathic molecule, like soap. Okay. 5. Compress the pin into a sphere that weighed the same as the pin. Would it sink or float?
It would float, right?
Nah, it'd probably sink. One thing is, like I said, you can float things in water that are heavier in water if you spread them out enough. You got to spread them out. So the mass of the pin is spread out. You know, the mass of an aircraft carrier is spread out. And the picture in the book of the spider walking on water, the water spider, its legs are splayed out, and you can see that each end, the tip of each leg is dipping into the water. So its mass is spread out. And that's the way you can float things that are heavier in water, but not if they're compacted into a tight ball. It's gonna sink. Okay. 6. As water is heated, hydrogen bonds between water molecules are broken. With this information, explain whether surface tension of water is greater at 20 degrees or 50 degrees. What do you think? Anybody?
Surface tension would be greater at 20 degrees.
Yeah, because there are more hydrogen bonds. So there's a direct relationship between the surface tension of water and the temperature. The greater the temperature, it's an inverse relationship. The greater the temperature, the lower the surface tension. And if you bring water to boiling, you break so many hydrogen bonds that they escape it in huge quantities into the air. And that, of course, that's steam, right? All right. Water is a solvent. Okay. So water, soluble in water? Yeah. This is like a control. and of course it's polar. Table salt, soluble?
Yes.
Yes. Is it? It's considered polar, but it's all. But a better thing to put here is ionic, because it's an ionic compound, so it's ionic. Does sugar dissolve in water?
Yes.
Yeah, yeah, that's kind of obvious. So that's polar. Does alcohol dissolve in water? Well, heard of mixed drinks? So alcohol is soluble. It's polar. But what about the mineral oil?
No.
No, it's not soluble in water. It's non-polar. That's the only one that's non-polar. So two, how can you decide whether a material is soluble? Like I said, you have it in lab. It's a powder or liquid. You don't know whether it's molecular formula. Put it in water. If it dissolves, it's polar. If it doesn't, it's non-polar. So it's easy. Three, interesting. After observing the solubility of each substance in table 4, what do all the soluble materials have in common, and how do they differ from the insoluble materials? Well, the only thing I think I can say is all the soluble materials are polar. The insoluble materials are non-polar. You could say the bonds, but not all the soluble materials have the same type of bonds. The salts have ionic bonds. The other things, the sugars and the alcohol, they have polar covalent bonds, and that's why they're polar. Oh, and well, and that's not actually that's answering number 4. Explain why polar substances are soluble and non-polar substances are not. They're either ionic, which always breaks apart in water, or they have some polar covalent bonds. So they got oxygen or nitrogen in them, and those two atoms make polar covalent bonds. So some people made an error in the molecular models lab when they said, "Oh, amino is a non-polar functional group." No, it's not. It's polar. They were all polar except for methyl. Okay. 5. What could you do to increase the rate at which the soluble materials become dissolved? What could you do?
You could stir it.
You could stir it. That's it. You could do that. What else could you do to it?
Heat it up.
You could heat it up. The molecules moving faster are going to dissolve faster. And 6, what could you add to increase the solubility of the insoluble molecules? Look at number 3. It's somehow put a polar covalent bond in there. You modify it somehow by putting a polar covalent, like butane. It's just 2 carbons and 6 hydrogens. It's insoluble. Add a hydroxyl group to it. You make ethyl alcohol. That is soluble. Okay, this is still going on in number 8. In table 4, predict the solubility and polarity of the substances. And so it's pretty much the same thing, but you got to do it. Okay, 2 sugars, soluble, right? And they're polar. Salts, calcium chloride and sodium nitrate. Now, here again, the nitrogen is making an ionic bond here. And so they break apart in water, so salts are soluble, and they would be ionic. These two are ionic. And you got two alcohols. Remember, all alcohols have hydroxyl groups. Hydroxyl groups, polar covalent bonds, so they're going to dissolve. So all alcohols will dissolve in water, and so they're polar. And of course, we get down to the two oils, insoluble, and so they're nonpolar. Okay, let's go on to part 4, question 1. Record observations. Okay, in the well is, we say it's water, it means distilled water. pH 7.0. Okay, B, you add hydrochloric acid to water. What happens to the pH? Oh, you have to just say up or down.
Down.
What?
Down.
Down, down. And then C, water and sodium hydroxide, up. That's a base. So you could say it gets higher or gets lower. It goes up or goes down. All right. Water, you put salt in the water. What happens to the pH?
Nothing.
Nothing. Why?
Salt doesn't have any hydrogen ions or hydroxyl ions.
That's exactly it. If it doesn't have any hydrogen ions or hydroxide ions, it doesn't break into those. It doesn't have any effect on pH. So that's 7.0. And then again with buffer, remember you can add buffer to pure distilled water and it stays at 7.0. Now, you got the buffer and you got water. When it says buffer, it means the buffer's in the water. You add a drop of hydrochloric acid. What happens to the pH?
Goes down.
Goes down, but how much? Just a little. It still goes down, but just a little. Not nearly as much as B. And same thing with G. You add a base, sodium hydroxide to the buffer. Goes up a little. And finally with H, buffer and sodium chloride. What happens to the pH? Nothing again. Remember how buffers work. You add HCl and lots of hydrogen ions are formed in these buffer molecules. They can soak up hydrogen ions. They take them and they make bonds with them. And that takes them out of solution. Same thing on the other side with sodium hydroxide. The buffer can provide hydrogen ions. The hydrogen ions bind to the hydroxide ions to form water. And that's how buffers work. Okay. And here, so down here is just answering the same questions we just went over. Which substances add to water because A to D lowered the pH? And is this substance an acid or a base? And so we've already gone over that. 3, with substance when added to water, A through D raised the pH. Is this substance an acid or a base? It's a base. And then 4, which substance when added to water did not change the pH? And just write down the letter of those. And then 5, compare the pH of beakers A and E. So A is just pure water, E is buffer. There's no change in pH. They're both 7.0. Then 6, compare pH of beakers B and F. So B is water and acid. F is buffer and acid. So the beaker without the buffer experienced a greater change in pH. And so the effect of adding acid to water compared to the effect of adding acid to buffer, you get a much smaller pH change with a buffer. And that's the whole purpose of a buffer. That's the whole function of a buffer. And 7 is the same thing, but in other direction, C and G. Adding sodium hydroxide to pure water, or adding sodium hydroxide to water that has a buffer in. Number 8, we already answered. Table salt is an ionic compound that dissolves easily. Why does the salt change the pH of a solution? Because it doesn't add hydrogen ions and it doesn't add hydroxide ions. Okay. We went to 5. Draw a picture of your beaker. Now the drawing question, you don't have to, don't worry about that. Also, the next one is the drawing. So 1 and 2, you don't have to draw it. But how does the density of the 3, how does the density of water change as its temperature increases from 0 to 4? So what happens to the blue ice as it melts? It falls, right? Well, what happens to the blue water that made up the ice as the ice melts? It drops to the bottom. So as it goes up, as it increases the temperature from 0 to 4, its density goes up. Wait a minute. Okay, 4, how does the density of water change as its temperature increases from 4 degrees to room temperature? What happens to cold red water as it warms up? So as it warms up from 4 to like room temperature, its density decreases. Okay, now here's a thing that I would like you to draw. Number 5. Sketch and label a thumbnail graph that shows the relationship between temperature and density. So thumbnail just means by hand, just draw. And so going back to the first lab, no, the second lab, which of these is the independent variable and which is the dependent variable? You got temperature and density. Well, what's dependent upon what? Is the temperature dependent upon the density, or is the density dependent upon the temperature?
Density.
Density. Sorry.
So is density dependent or independent?
It's dependent.
Dependent, yeah. So put temperature on the x-axis and density on the y-axis. And what's this curve going to look like? An inverted V. That is, if you have low density at the bottom and high density at the top, because remember, water is most dense at 4 degrees. All right, see that? Okay. 6. Relationship between the number of H bonds and the temperature of water. Again, temperature being the independent variable, H bonds dependent, and as the temperature goes up, the number of hydrogen bonds decreases. So it's going to be an inverse relationship, negative slope. So as you sketch so another thumbnail, showing the relationship between temperature and the number of hydrogen bonds, and you're going to have a straight line with a negative slope. The hotter temperature is, the fewer hydrogen bonds there are. And then explain why ice cubes float. Because the water molecules are farther apart in ice than they are in liquid water. And it's a bizarre property. I don't know why that happens. Somehow, as water cools down, it gets colder than 4 degrees, the water molecules actually start to get farther apart. And I'm not sure why. I think it has to do with something with the stability with the crystals. It has to form a crystal, and the water molecules have to be farther apart to make that ice crystal. And so they move farther apart, and it's less dense. That's why ice cubes float, less dense than liquid water. And what effect does this have? What biological consequence does this have? It has a big, ecological consequence, because since ice floats and is less dense, bodies of water freeze from the top down rather than from the bottom up, and that protects all of the organisms in the water, because a lot of them are down below in the mud at the bottom, and if that froze first, for most living things, freezing is death. You freeze something, you're going to kill it. A couple of exceptions to that. But what are you talking about? You can freeze human embryos, and and then you throw them out and use them and implant them in a woman. Well, that's artificial freezing. They have to treat those embryos specially to be able to freeze them and not kill them. Right. So that has big consequences. Oh, and one other, I'm almost forgetting one other property of water I want to mention. And we used to have an experiment to show this, but it was taken out of this for some reason. That's called capillarity. And that's the ability of water to defy gravity in small tubes. What we would have would be these small glass tubes. Each one would have a different diameter. There'd be one with bigger diameter, smaller and smaller diameter, and we'd have them put into green water. And then you'd be measuring the height of the column of the water in each tube. And the relationship is, the smaller the diameter of the tube, the higher the column of water. And so it would defy gravity because of its cohesiveness and adhesiveness, because the water is attracted to the polar nature of the inside of the glass, and it's attracted to other water molecules. So as it moves up the glass, because it's attracted to it, it pulls along other water molecules with it, and they move up. And you can only raise a certain volume of water that way against gravity. That volume of water in the tubes that have a larger diameter is smaller and squatter. But in smaller ones, the same volume is squeezed. So it's taller and taller, narrower and narrower, and taller and taller. It's the same volume of water. So how is this biologically relevant? Well, not in us, but in plants, it's pretty relevant, particularly in trees. Now, we have a pump that pumps fluid throughout our body. You know, trees don't. They don't have a pump. But they need to get water to the leaves at the top of the tree because water is used in photosynthesis. CO2 and water plus sunlight make sugar. So you gotta get water up there. How do you get water up there? Well, just underneath the bark are these tiny tubes. The tubes with a microscopic, diameter of the opening, and they're called xylem tubes, and they carry water. Now, where does a tree get its water from? It gets it from the roots, right? So it's got to take it from the roots and put it up to those leaves. And capillary is one way it gets it up there. Now, there are other forces at work here that we don't have to get into, but that's an important force for life on Earth, for trees. Okay, it's called capillarity. All right, I think this is the last thing, part 6. We still got an hour and 15 minutes. So draw a picture comparing the cut faces. Another draw a picture you don't have to do. But you'd see that, remember that the cut face of the small cube was pink all the way through. The cut face of the larger cube, only a thin band of pink on the outside. It's all milky white on the inside. Right. Shows that diffusion was faster in the smaller cube because it has a much larger surface area to volume ratio. And that's how, number 2, that's how you can tell how far the sodium hydroxide diffuses into the tube. How thick is the layer of pink? Is it all the way through, or is it just on the outside? In 3, we already said this, in which tube did this sodium hydroxide reach the interior tube more quickly? The smaller one. And I think the last question is why? Why did it diffuse faster in the smaller cube? Because the smaller cube has a much larger surface area to volume ratio. So that's it. That's all the questions. So any other, so need to get that to me somehow. Usually people ask how, you get to be the same way you got the other ones to me. You could make a extension attachment to an email. Some people say oh, I'll take a picture of it. Well, you can do that also, but that makes a little more work for me because usually when people take pictures and send it to me, the picture is sideways. Either I got to read it like this, or I have to save it to the computer, put it into the viewer, and then there's a little arrow on the viewer that makes me rotate it so it's right side up. Or you can just hang on to it and turn it into me when we go, next week and the following weeks, I hope, we'll be going back onto campus. So you can just hang on to it and turn it into me there if you want.
So all the labs we haven't turned in yet, we can give to you when we see you on campus?
Yeah, if you haven't given it to me, you can give it to me when we get on campus. Yeah, that's fine. That's fine.
Okay, they won't be considered late though, right?
No, they won't be considered late.
Okay, thank you.
Okay, so if there's no more questions or comments or problems or anything, I'm gonna end the meeting a little early. We got until 12:45, but we're pretty much done. And I'm going to stop recording now.